In the last video, we talked about gene regulation in prokaryotes, and now we're going to talk specifically about gene regulation in eukaryotes. It's first important to understand why there's a difference in regulation, and that can mostly be attributed to the complexity of the DNA. So, for example, if you compare prokaryotic DNA, that typically exists in the form of an operon, and so it's closely sequenced genes found very close together on a part of DNA. With eukaryotic DNA, we have several introns that are non-coding regions, and so it's going to be a little bit more of a complex process. We're going to focus on 5 different levels of gene regulation in eukaryotes. Specifically, we're going to talk about chromatin structure, transcriptional control, so how do we control transcription occurring, post-transcriptional control, so what happens after transcription, translational control, and post-translational control. This diagram here is just showing how the central dogma of biology, essentially, DNA being replicated, undergoing transcription to mRNA, and then mRNA turning into protein through translation, and all of those kind of checkpoints, if you will, to regulate gene expression. So we're going to first focus on chromatin structure. We've mentioned this when we talked about mitosis, but remember that DNA is very organized. We have DNA that can be wrapped around individual histones, which are proteins, very similar to strings on beads on a string. When we start clumping those together, you can start forming things that are called nucleosomes, which then supercoil, and then you can get chromosomes. Chromatin structure can have 2 main forms. We can have euchromatin and heterochromatin. Euchromatin is the chromatin that exists in cells that is very loosely packed. And so that's the typical beads on a string. If we look at this diagram over here, all of this white looking material, that's euchromatin. It's very loosely packed. Sticking out on the nucleosomes of those euchromatin are these little tails that stick out off the histone proteins, and they contain these acetyl groups. It's because of the presence of that acetyl group that the euchromatin is going to be the DNA that is typically going to be transcripted and then translated. This is contrasted to heterochromatin, which is very densely compacted, and this is not going to produce anything because transcription cannot occur. The histone tails from the nucleosome contain methyl groups, and that methylation, or the presence of the methyl groups, will actually prevent transcription from occurring in that type of DNA. In that diagram, it's going to be all of the black area that's pushed right up against the nuclear envelope, or excuse me, the cell membrane. The second type of control that we have is transcriptional control. So how do we control transcription? Well, we can essentially control transcription at how fast it occurs. We're going to talk specifically about how this process could work. So we have these things that are called transcription factors, and that can be seen as these kind of purple dots here. And they typically aid in RNA polymerase binding to the promoter region. Once that RNA polymerase binds to the promoter region, like you can see here, we then have these transcription activators, which are these little yellow structures that attach to these areas called enhancers. When that process happens, the activators on the enhancer side bends over and reaches where we have the promoter region. Those two, the activators in the RNA polymerase, are attached through something called mediator proteins, and then transcription can occur. So why do we talk about this? Well, we can actually, or our cells have developed a way to moderate how quickly these transcription factors and transcription activators attach to their to the specific regions on a gene that needs to be transcribed. A post-transcriptional control exists in cells, so pretty much alternative splicing. So we've talked about processing mRNA or splicing out introns, and there are various options when this occurs. So in this diagram here, you have your original DNA strand, which has 5 exons, which need to be pushed together to get sent out. So we have one method where all 4 exons are spliced together. The introns, or the non-coding segments, were eliminated. So then you can get protein A here, for example. You can do a different kind of splicing where you actually cut out one of the exons, so a coding region, depending on what kind of protein you want to make. So in this example, they actually spliced out the third exon. Whatever protein this cell was trying to make, it didn't need that section, so it got rid of it along the introns. And then another option could be something like you see here, where the fourth exon was also cut out along with the other introns. And so this is important in that different cells are going to need different proteins. And so we need to have a post-transcriptional control or some way of getting rid of the segments that that cell does not need. We're moving on now to translational control. So how do we control translation? And typically this is just going to be talking about the 5' cap and the poly A tail on the 3' end. So if one of these does not exist or it is damaged, it can actually lead to these exons or the coding region that you need to be harmed in a way. And so the translational control in eukaryotic cells are going to be the addition of that 5' cap and the poly A tail. Post-translational control exists in our cells as well. So this is actually an interesting process. When those proteins are made, or that long polypeptide chain is made and it undergoes all of its folding, we then have a protein. Well, sometimes we don't need the protein to stick around very long. We only need it for a very short amount of time. So what happens is that these proteins are tagged. And this tag is going to allow an enzyme known as protease to recognize it. Protease is an enzyme that is found in something called a proteasome. The proteasome contains proteases, which can then break down that tagged protein. So this allows us to control how long a protein stays in existence within a cell. That's the end of this video. There's a ton of information. We're going to talk a little bit more about it in class, but make sure you follow along in your book in chapter 11 for eukaryotic gene regulation.